Dear friends, welcome to the biochemistry lecture. Today we learn about more about the absorption spectrum and emission spectrum and what is its relation with each other. Absorption spectrum is a kind of spectrum which will obtain, or that kind of pattern of spectrum which is obtained after transmission of a light from solution which containing a particular kind of substrate, etc., and that absorb pattern is known as absorption spectrum. Here you will observe that light is passing through a cuvette and the substrate is present here in the cuvette. That substrate molecule or that substrate concentration absorbs some amount of or some wavelength of light, and when it pass through the prism we will get a particular spectrum. In this spectrum, if we observe this spectrum with the continuous spectrum, we find some gap between the continuous spectrum, and these gaps are known to be absorption spectrum because this wavelength particularly get absorbed due to the presence of that substrate. If we plot this graph against absorption and wavelength, we will get a kind of peak here where the gaps are present. That means particular that wavelength of light get absorbed by the molecule. This kind of pattern is called as absorption spectrum. Then question arises, what is the emission spectrum? You all know that when we give energy to a substrate molecule, it will charge up and emit some kind of radiations. So here is the solution which containing the substrate, and we are providing energy to it. Because of excess of energy, that absorbs some kind of energy and emit or emitted another kind of energy or some amount of energy. That emission is trapped under prism, and then when it is analyzed on the screen, then we will get a pattern where there is some continuous lining and gap is much more. So these type of spectrum is called as emission spectrum. Suppose we provide the heat or energy to the electron, the substrate electron from ground state will goes into the excited state, and then when in excess amount of energy, it will releases some amount of energy so that it again reaches to the ground state to achieve the configuration, to achieve the stability, and during that process it emits some energy, and that emitted energy will give a spectrum. That is what we called it as emission spectrum, and that emission spectrum is nothing but the what we called it as absorption spectrum is the negative of the emission spectrum. Or if you compare that, where the gap present here, exactly just in near the gap or on the gap, here is the filling. So it is called as the absorption spectrum is called as the negative of the emission spectrum. So that is what we call it as the absorption spectrum and emission spectrum. Both are the either sides of the coin, and the absorption spectrum and emission spectrum both will give idea about the nature or chemical composition of the substrate present here, because the substrate is actually responsible for this kind of various spectrum. And studying the absorption spectrum and emission spectrum, we can identify the particular substrate which is present here, because you all know that various kinds of substrate have its own chemical structure. So the absorption spectrum and emission spectrum for a particular substrate is specific for that substrate, and with the help of that we can identify the substrate which is unknown to us. This study is carried out under what we call it as the branch spectroscopy. Spectroscopy is a branch which studies the absorption spectrum as well as emission spectrum, and it will give idea about the nature of the substrate. Thank you.